How about the co-scientist? What happens when AI is given complex data and a scientific goal? Because I'm pretty sure if all I did for my PhD is like collecting data, which could have been done by a simple robotic arm. Yeah, so if that thing was given the data I collected during like 4.5 years, it definitely could have written the dissertation, so my PhD thesis and papers and stuff. I'm pretty sure of that. Yeah, currently we are on NotebookLM, so we use that as an example. It can accelerate discovery, but not without risk. I don't know if it's copyrighted. One thing it's good at is getting rid of copyrighted material. We'll obviously give it our own website as well, because that's essentially what we were doing. It's not allowed to be crawled by AI. Yeah, it's like a university website. And that's okay, so we can get rid of it. Yeah, AI is starting to act less like a tool and more like a research partner. That's right. Should I give it my thesis as well? I mean, the data, so I was saying, you probably, if you gave it the data set, it will able to write your PhD dissertation. So the university doesn't want their website to be crawled by AI. Well, how are they going to appear in searches and things? That's an interesting question, but we're not going to cover it here. It's funny, so they don't allow crawling on the website, but that article does have a note at the bottom saying the author used an AI tool to assist in writing this article, which was carefully reviewed with input from all teams. Okay, yeah, we know how's that normally done. No one actually reads anything. The technology is moving fast. The question now is whether frameworks for academic oversight can keep pace. Well, obviously not, but that's also something we don't care about. Yeah, can we summarize the technical aspects of the article? Get rid of any names, of any personal names. Company names like ChatGPT, OpenAI, whatever is fine to keep, but get rid of any personal names. Yeah, making sure the summary is unique and not copyrighted in any. So essentially rewrite bits, don't just copy paste.
The article describes a shift from traditional prompt response AI systems to more autonomous co-scientist architectures designed to handle end-to-end research workflows. Instead of producing a single answer, these systems decompose a high-level research objective into many intermediate steps, including literature retrieval, dataset interrogation, statistical analysis, and synthesis of results. A single run may involve hundreds of chained operations executed over several hours, effectively mimicking parts of the iterative scientific method. Technically, these systems combine large language models with tool use capabilities and orchestration layers. They can query public scientific databases, integrate heterogeneous datasets, and apply analytical methods such as pattern detection or prioritization algorithms. Their strength lies in rapidly cross-referencing diverse sources, linking experimental data with external knowledge bases like protein databases or genomic repositories, and generating structured outputs such as ranked hypotheses or candidate biomarkers. In data-heavy scenarios, the systems accelerate early-stage research tasks. They can assist with pre-processing steps like exploratory data analysis, identifying correlations, and surfacing latent patterns across large datasets. This compresses what would normally be weeks of manual exploration into a much shorter time frame. However, they do not reliably handle methodological decisions on their own, such as defining variables, treating missing data, or managing statistical validity. These steps still require human intervention and domain expertise. In literature-driven tasks, the systems can aggregate and summarize findings across large volumes of publications, producing gene lists or variant interpretations based on existing research. This is particularly useful in fields where the volume of published work is difficult to track manually. However, limitations include incomplete access to restricted publications, occasional factual inaccuracies, and unreliable citation mapping, where reference sources may not actually support the claims made. Across use cases, a consistent issue is the generation of plausible but weakly supported conclusions. The systems can produce hypotheses that appear coherent, yet rely on insufficient data or misinterpreted signals. This is especially evident in complex biological datasets where inferred mechanisms may not hold under closer statistical scrutiny. As a result, validation becomes the primary bottleneck. Users must rigorously evaluate outputs to distinguish meaningful insights from noise or speculation. Prompt design plays a critical technical role. Broad prompts encourage exploratory and novel outputs but increase the risk of error, while tightly constrained prompts improve accuracy at the expense of creativity. Effective use, therefore, involves iterative refinement of instructions and continuous oversight of intermediate results. Overall, these AI systems function best as accelerators for hypothesis generation and data exploration rather than as independent research agents. Their integration into scientific workflows shifts the technical burden from performing analysis to supervising, validating, and interpreting machine-generated outputs.
Can you find like the opposite view of where there was someone is convinced, a professor or something, who is saying that it can do valid research, like out of the box? Can we find the reference of a less critical claim where a professor or someone says how AI tools can do research successfully, so could potentially write and submit whatever a PhD thesis and things like that, potentially without supervision or with minimal supervision? Yeah, not sending it to ChatGPT. We want to pop it into, actually we could. So let's use the search now. It's interesting that original article by the university is not crawlable by AI, which kind of contradicts some of the claims the article is making in terms of accessibility. Student booted from PhD program over AI use. But we find if the article itself was written by AI and just made up. University of Minnesota. It says PhD program, but all 4 faculty graders of his exam expressed significant concerns that it was not written in his voice. Can AI write your PhD dissertation for you? Well, I'm convinced it can, not because it will be high quality, but because the peer review process is broken. 4 days trying to get OpenAI new tool Deep Research to research and write a complete dissertation. It's questionable how original this AI-generated dissertation is, even though the simulated thinking it presents is quite impressive. To get a sense of this, I turn to Deep Research as to assess the originality of the completed work. Yeah, it's not only that you can use the AI tools to write your dissertations, you can also use them to assess it as well. Give it originality score of 70%. Okay. Yeah, let's get rid of it. Because it's somebody's personal take on things. Okay, now this is interesting. It's in some Advancing Digital Health, Open Science, blah, blah, blah journal. Evaluating the potential and accuracy of ChatGPT 3.5 and 4.0 in medical licensing and in training examination, systematic review and meta-analysis. No bias, conflicts of interest non-declared. Okay, we'll keep that one. Yeah, obviously, yeah, for a human it's like impossible. Well, first of all, you have to read the whole thing. How much money does AI save when writing papers? With AI scientist version 2, generating one paper costs about $20 to $25. Experiments. $15 to $20. Writing, $5. Experiments. Okay. Human researchers face formatting cost median for $500, open access free $4,000 plus labor, often totaling tens of thousands of dollars. However, 2 of 3 AI-generated papers were rejected, so the cost-quality trade-off should be carefully weighted. Are they trying to submit them as well? They're submitting papers where synthetic data meets AI science. So they're generating synthetic data, writing a paper, submitting it for publication. This was done in Korea. Yeah, I'll keep that one. And we have last one is archive. So open source, open access without publication. I'll just check it out if it's present. AI scientists fail without strong implementation capability. Sounds like quite a biased title. I'll keep it because it's a supposedly like written, at least written like a scientific paper. Yeah, there's some issues with formatting. You can get the PDF. It's from China. We'll keep that one. So we'll import those.
BioniChaos serves as a comprehensive digital hub that integrates biomedical engineering, neuroscience, and educational technology. The platform offers a diverse array of interactive simulations and data analysis tools designed for researchers, students, and healthcare educators. Users can explore specialized resources ranging from EEG and ECG signal processing to complex models of human physiology and robotic prosthetics. Beyond medical data, the site features artificial intelligence demonstrations, optical illusion viewers, and physics-based games to foster engagement with scientific concepts. These web-based applications utilize real-time visualization and auditory feedback to simplify intricate topics like quantum mechanics and magnetic resonance imaging. Ultimately, the collection aims to democratize biomedical data by providing accessible, browser-based environments for technical learning and experimentation.
Okay, so that's BioniChaos. We have it included in the sources.
The provided sources explore the emergence of AI research assistants and autonomous agents that are transforming the scientific life cycle. These computational tools utilize large language models and machine learning to automate complex tasks such as literature synthesis, hypothesis generation, experimental design, and manuscript drafting. While systems like Sakana AI's AI Scientist and Google's Co-Scientist aim for end-to-end automation, current evaluations reveal significant hurdles involving hallucinated results, logical inconsistencies, and a lack of deep critical analysis. Despite these technical flaws, the technology offers a paradigm shift toward human-AI synergy, significantly reducing the time and cost required to produce research. The rise of these tools has prompted the creation of AI-specific journals and specialized markup languages to manage the growing challenges of academic integrity and data quality. Ultimately, the sources suggest that while AI cannot yet replace human insight, it is becoming an indispensable accelerator for scientific discovery.
Okay, so that's the summary of the sources. It's not actually going into any detail. Okay, we'll keep going on that one. We will ask it to do like an audio overview, slide decks, whatever. Mind map could be quite useful. No, flashcards. I don't know what flashcards are. Infographics sometimes are useful. So essentially, quizzes. Yeah, everyone likes a good quiz. Not sure about reports. Probably no need. Unless we're putting it as a blog on the website. But then, yeah, I don't think blogs are necessary anymore. Probably don't need them. But we will give it a go. Have a look. Mainly relying on the audio overview. And it can also do a video overview. So we'll try that one out as well. So we'll actually, it takes some time to generate. It's actually, I don't know if it will time me out, because obviously, yeah, there's a limit of how many notebooks you can have, but supposedly there is no usage limit. I don't know. Yeah, so we'll generate the audio overview, a brief critique, debate between two hosts. Well, debate, yeah, in this case would be nice. A short, default, yeah, English. Any comments? No. Just generate a debate to a slide deck, present a detailed deck, comprehensive deck with full text and details, perfect for emailing and reading on its own. Clean visual slides with key talking points to support you while you speak. Okay, let's do a presenter. A video overview. Explain a brief. Yeah, I'll do a brief. So video, slide deck, audio. Yeah, I like the infographics. Because essentially what we're doing like interactive. Well, to get a simulation going, you essentially need an infographic. Yeah, landscape. Standard detailed. What's the difference between concise and detailed? What? Just go for standard, see what it does. quiz. We can do a quiz together, see if it actually makes sense. But luckily we have one that we prepared earlier. This is on the title is not great, but it's essentially on BCI, on brain-computer interfaces. For this one, we already generated quite a lot of things. Let's look at the video. Wait, where is the video? Yeah, the graphics. That nonsense. Yeah, we'll play it. It's only 6 minutes, 7 minutes. Yeah, increase the speed by 25%.
You know, for decades, the idea of connecting our minds directly to machines felt like something straight out of science fiction. But today, it's one of the most exciting and one of the most challenging frontiers in all of science. Just think about it for a second. What if you could control a robotic arm, communicate with the world, or even get your senses back just by thinking? No keyboard, no Siri, nothing. Just a seamless, direct link between your thoughts and the digital world. Well, that is the incredible world of brain-computer interfaces, or BCIs. The whole goal here is to create a direct information highway between the human brain and a computer, completely bypassing our muscles. And this isn't just about giving us superpowers, it's about restoring what's been lost. And the potential applications are just staggering. I mean, we're talking about giving movement back to people who are paralyzed, helping the blind see again, helping the deaf hear, even finding new ways to treat tough conditions like epilepsy and depression. This tech could literally change millions of lives. But there's a huge hurdle. The real problem, the central challenge, is right here at this connection point. How in the world do you safely connect our incredibly soft, squishy, delicate brain tissue to the hard, rigid electronics you need to read its signals? Well, it turns out the brain doesn't just welcome this new piece of tech, it actively fights back. The moment we implant an electrode, our brain launches this fierce defensive response. It basically treats this amazing, revolutionary technology like a hostile invader. And what's really wild is the sheer scale of this mismatch. Okay, so brain tissue is one of the softest materials in our entire body. Now, compare that to traditional electrode materials like silicon or metal. They're thousands, sometimes even millions of times stiffer. This massive physical difference is basically the starting pistol for everything that goes wrong. And you might think this is some unique, mysterious brain thing, but it's really not. It's the same fundamental foreign body reaction your body launches if you get a splinter in your finger. The brain recognizes there's a hard, rigid object inside it that just doesn't belong, and it immediately goes on defense to protect itself. So let's break down exactly what happens. First, just putting the electrode in causes an injury. That's step one. Second, the brain's own immune cells, these things called microglia, sound the alarm. Then, over the next few weeks, other cells rush to the scene and start building a thick wall of scar tissue around the electrode, trying to quarantine it. And finally, that scar acts like an electrical insulator, blocking the very signals we're trying to read. The connection is lost. And what's the end result of all this? Well, because of this intense immune response, many of these cutting-edge devices just stop working. They fail in as little as 6 months to a year. For a technology that's supposed to be a permanent solution, that's a massive, massive roadblock. So how do we fix it? Well, researchers are tackling this from two main angles. The first one is a pure engineering challenge. The thinking goes, if the problem is a physical mismatch, then let's just build a better, more brain-friendly bridge. And the main idea here is actually pretty simple and elegant. Make the electronics act more like the tissue they're sitting in. You want them to be softer, smaller, and way more flexible. That way you minimize that initial injury and the long-term irritation. So one way to do that is to take traditional materials, like gold, but completely change their form factor. I mean, look at this thing. This is an array made of ultra-thin gold, and it's so flexible it can basically hug the surface of the brain, which dramatically cuts down on the mechanical stress. Okay, another strategy is to use materials that are just soft to begin with, stuff that's naturally more like the brain. This here is a membrane made from a graphene hydrogel. Hydrogels are mostly water, they're super flexible, kind of like Jell-O. They're much more like brain tissue, which helps calm down that inflammatory response. And we can even zoom in further, all the way down to the nanoscale. These are pretty cool. They're called whiskered gold nanosheets. By creating these tiny, complex, textured surfaces, you increase the surface area for a better signal, and you create a much more inviting place for neurons to get nice and close. It improves the connection right at the cellular level. But engineering alone might not be enough. So that brings us to the second big approach. Instead of just trying to be a quiet, invisible guest inside the brain, what if the implant could actively negotiate a truce with the brain's immune system? This is where we shift gears from physics and engineering over to biology and chemistry. The idea is to use the implant itself as a tiny drug delivery device that basically tells the brain's immune cells, hey, it's cool, stand down, this guy is with us. And as this table shows, we can get really precise. You could go broad, using a general anti-inflammatory steroid to just calm everything down. Or you can get much more specific, using special coatings on the electrode that stop that insulating scar from even forming. You can even block the specific molecular signals that kick off the whole inflammatory chain reaction in the first place. And this quote from the researchers really gets to the heart of where the whole field is right now. They say that better physical design is absolutely critical, but it's not going to be the whole answer. To get these things to truly work long term, you're going to need a combination of smarter engineering and smarter biology. So where does all this lead? What's the ultimate vision for this technology, beyond just getting the signal to last for more than a year or two? Well, the ultimate goal is a complete paradigm shift. We need to move away from just creating a biocompatible implant that the brain just kind of tolerates and move towards a bioactive implant that the brain actually partners with. And that brings us to this really key concept, bioactivity. This isn't just about being invisible to the immune system. This is about the implant actively doing good things. Imagine an electrode that doesn't just listen to signals, but also releases chemicals that encourage damaged neurons to repair themselves, or that encourages new neural connections to grow towards it, making the bonds stronger over time. And that's really the profound question this research leaves us with. What happens when the line between a piece of technology and a medical therapy completely blurs? When the devices we implant don't just bridge a gap, but become a regenerative part of us, helping our own biology to heal and integrate. Solving that's the key to unlocking the full, incredible promise of connecting our brains to the world.
Okay, so that was pretty good. There's obviously some copyrighted material in the video. I don't know where they got it from. Yeah, this is a slide. Yeah, that's a slide that actually obviously came from somewhere. Is it one of the, I assume, ideally it will be in one of the sources. It should have specified that on the slides. It should have said where it's coming from. It didn't. So NotebookLM is plagiarizing. So surely we would not be showing this on our YouTube channel. What are you building? Is this open source? Yes. What are we building? We're building this website, BioniChaos.com. So essentially everything we do in live, in the live streams, ends up on this website. And yes, it's all open source. Just go F12 and grab the code. But yeah, don't get too excited. Yes, if you go F12, this all should be JavaScript. Yeah, just click on that. I don't know if it was already selected. I don't know why it's not showing the code straight away. And the code is there. So in this simulation, it's just under 800 lines of code. You can just grab the code from here. All the JavaScript is in the same HTML page. So all the calculations, trajectories, all the functions, dysfunctions, pun intended. The sound generation, synthetic sound. Yeah, and all of it based on real science. So the paper or whatever this is based on should be at the bottom somewhere. If there isn't, that's because whatever bot made this forgot to include a reference. But yeah, originally it would have been based on some scientific paper. Well, yeah, and it's forgetting to include references like like this NotebookLM generated a video. They ripped some slides, some graphics from one of these papers probably that we included. I'm not going to go find it. But yeah, essentially makes it not usable because it forgot to include the reference to the thing. So is this open source? Yes, it's open source. How big is BioniChaos? Very big. So yeah, if you do have comments, especially after checking the website, yeah, close the F12 whatever. Yeah, if you check the site, you have any specific comments, questions, complaints, more than happy to take those. Anyway, I was saying NotebookLM, generated this, generated a gazillion of things. Obviously, they are quite useful, I would say, all of them. Yeah, generated the blog post, explainer, whatever flashcards are, deep dive, audio, video, quiz. Yeah, generated a lot of stuff. We checked the video. The problem with the video is that there is a copyrighted thing in it, so we can't actually use. Yeah, this one, for example, just ripped a whole slide, a chart from a paper, and didn't specify where it took it from. It should have mention it on the slides in the video somewhere, probably both in the voice and on the slide as a reference. The visuals are actually like legit. Let's go over the visuals real quick. Yeah, that one is nonsense, but I mean, it's fine. It's just the title thingy. Yeah, this one. So here you've got a robotic arm, a brain thing. So yeah, when you see something like that, who is actually doing the drawing? Is it the neurons? The AI must be connected to AI, obviously. Who's doing the drawing in that case? Yeah, we don't know. Yeah, those images are pretty generic. That one's an interesting one. There's a lot to unpack there. Yeah, this image, yeah, I get it. So you have this like rigid electronics that we're putting in the brain, and that's what the video is talking about. So it's actually, the audio is actually makes a lot of sense. Yeah, the video does make a lot of sense. Yeah, the main problem I have with it is that it just ripped graphics from somewhere and didn't mention where it came from. So we're not gonna share it. Well, obviously we already did, so I don't know. Yeah, but I'm mentioning, yeah, it's unknown source. It seemed to not actually matter. Like, I don't know why. It shows that that those graphics, it's really packed. I mean, it could have just like replaced the graphics. I wonder if you can, where is the actual prompt for the video? Good video, bad video. Well, you can download the video, that's fine, but you can't change the video, can you? I don't know.
Amazing, revolutionary
Amazing
is the sheer scale of this mismatch. Okay, so
yeah, it's using some big words that I don't like. We could tone it down a notch.
They're thousands, sometimes even millions of times stiffer. This massive physical difference is basically
so talking about the rigidity of the electronics, the electrode arrays that are placed in the brain, and the problem with that, essentially killing the tissue. Yeah, that graph is actually, well, surprisingly, it makes a lot of sense in the scale. I mean, I understand it. I understand it's trying to have like the same form factor, whatever, like the size, showing you. Yeah, it actually makes a lot of sense. So I like the logic it's using.
the starting pistol for everything that goes wrong. And you might think this is some unique, mysterious brain thing, but it's really not. It's the same fundamental foreign body reaction your body launches if you get a splinter in your finger. The brain recognizes there's a hard, rigid object inside it that just doesn't belong.
It's why this thing just yeah, it's kind of, I don't understand why it's sharp. Why is it sharp on the other end? And why these things are different color, whatever they are. Yeah, so it's like eliciting a biological, like an immune response. But those things could have been just the same color, whatever.
And it immediately goes on defense to protect itself. So let's break down exactly what happens. First, just putting...
Phew, this is a bit packed. Why those things wearing a bra? And that thing is a total mess. What's going on? What's happening there? Mueller?
The electrode in causes an injury. That's step one. Second, the brain's own immune cells, these things called microglia, sound the alarm. Then, over the next few weeks, other cells rush to the scene and start building a thick wall of scar tissue around the electrode, trying to quarantine it. And finally, that scar acts like an electrical insulator, blocking the very signals we're trying to read. The connection is lost. And what's the end result of all this?
What is that? Anyway, yeah, I understand what it's trying to say. I don't understand why these things are wearing a bra or whatever it is. Upside down bra. There's an alarm clock, whatever. That's messy. I understand what it's trying to say, so it's actually, it's good overall. I don't know why the graphics are so complicated, but yeah, it's to do with like the encapsulation. So whatever you insert, would be the tip of the electrode, and it gets encapsulated by scar tissue. That's what it's talking about. Could have done it in a better way. I'll blame it. I call the complexity on the sources. The sources are probably don't explain it properly as well, because whatever scientific papers tend to overcomplicate things. What is the benefit of modifying electrode surfaces with nanostructures like nanoparticles? Yeah, well, You can interact with the tissue better, increasing surface size. It increases the interface surface area and improves contact with neural tissue. Did I answer it correctly? What is the first and most critical event that occurs during the implantation of neural electrodes? Disruption of blood-brain barrier. Anyway, it's a great tool for learning. If you are in med school and stuff and you're not using NotebookLM, I don't know what you're doing. Right, so it will remember your progress as well, which is nice. The audio should be nice, especially because it has an interactive mode, so we can interrupt and talk to the thing. A seamless neural interface. What's that? A slide deck, is it? Slide deck. What on earth? That number sounds crazy. It's kind of similar to what you had in the video. Kind of more generic. Yeah, it's taking those images. You have to actually mention the source if that's a real image. Or did it looks like a real image? That one looks fake. You know, as tools like this become better, we'll probably won't even know what's real, what isn't. What's that number from?
Usually when we think about the future of technology, we imagine like these incredibly clean lines.
Right. Very sterile.
Yeah, precision. We picture something straight out of a sci-fi movie. A flawless metallic chip syncing perfectly with the human mind.
That is the popular image for sure.
And the promise of brain-computer interfaces, or BCIs, is exactly that. I mean, we are talking about restoring movement to paralyzed limbs, treating severe depression, and maybe one day unlocking human augmentation.
Which is incredible to think about.
It really is. But there is a massive biological elephant in the room that almost never makes it into the tech headlines. The reality is that the human brain aggressively, almost violently, hates having foreign objects introduced into it.
It really does. It acts like an uncompromising biological bouncer.
Big bouncer!
Yeah, because we tend to view interfacing with the brain as a pure software problem, right? Assuming it's just a matter of reading the right neural code.
Just plugging in a USB drive.
Exactly. But before you can even begin to translate those signals, you have to survive the physical reality of the brain itself.
And that is exactly our mission for you today in this personalized deep dive. We are exploring the absolute cutting edge of brain-computer interfaces.
There is so much happening in this space right now.
Oh, totally. And to do that, we are synthesizing some incredible research we have on the desk today. There's a fascinating materials science review detailing the newest physical electrode innovations, alongside an immunological study mapping out exactly how we can stop the brain from chemically attacking these implants.
Both of which are crucial pieces of the puzzle.
Right. Okay, let's unpack this. Before we can talk about the engineering solutions, we really have to understand the crime scene. Why does the brain reject these electrodes with such extreme prejudice?
Well, it fundamentally comes down to a mechanical mismatch. We have to look at the physical properties of what we're actually putting inside the skull.
So the actual hardware.
Right. Traditional electrodes are constructed from rigid metals and inorganic semiconductors. You're looking at materials like silicon, gold, or platinum.
Very hard materials.
Extremely hard. And on the other side of the equation, brain tissue is one of the softest, most fragile tissues in the entire human body.
How fragile are we talking?
To give you a concrete sense of it, the research notes that neural axons, the long, delicate cables that neurons use to transmit signals, will literally break at just an 18% strain.
18%? That's nothing.
Barely any force at all.
So just to visualize that mechanism, it's like trying to stick a dry piece of spaghetti into a block of soft tofu without damaging the structural integrity of the tofu.
That is a perfect way to picture it.
I mean, you can be as careful as you want during the initial insertion, but the moment that tofu shifts, the spaghetti is going to carve a path of destruction.
That analogy hits the physical mechanics perfectly, because the brain is not a static environment. It constantly pulses with every beat of your heart. Yeah, it expands and contracts slightly with your respiration, and it shifts inside the skull when you move your head.
So it's always moving around that rigid spike.
Exactly. When you have a highly rigid pin sitting inside that soft, dynamically moving environment, the discrepancy in flexibility causes continuous micromotion trauma.
Micromotion trauma?
Yes. It is microscopic shearing and tearing of the cellular environment 24 hours a day.
Let me pose a logical hypothesis here. If the primary issue is just a physical mismatch, like a rigid object in a soft environment, shouldn't the tissue eventually adapt?
You would think so.
Because, like, if someone gets a piercing, the surrounding tissue gets inflamed for a bit, but then it builds a neat little pocket and heals around the metal. Why wouldn't the brain just do the same thing and settle down?
You'd reasonably expect that, but the central nervous system doesn't play by the same physiological rules as the rest of your body.
Really?
Yeah, the brain is an incredibly isolated, highly protected fortress. The moment an electrode penetrates the tissue, it mechanically ruptures the blood-brain barrier.
And that's a big deal.
Huge. That breach is the ultimate biological alarm bell. Within literally minutes of the blood-brain barrier breaking, the brain's resident immune effectors, called microglia, detect the damage.
Wait, what are the microglia actually doing in those first few minutes?
They are sensing chemical distress signals, like ATP leaking from the crushed cells. Normally, microglia sit in a resting branch state, just monitoring the area.
Like little security guards on patrol.
Right. But once they detect that leaked ATP, they rapidly undergo a morphological change.
They change shape.
Exactly. They pull in their branches, become amoeba-like, and physically crawl toward the site of the injury. They shift into a highly pro-inflammatory state.
Wow, so they go on the attack immediately.
You do. And over the next few hours and days, they begin releasing cascades of chemical signals, specifically interleukins, which call in the heavy machinery.
The heavy machinery.
Yeah, the astrocytes.
So the astrocytes are like the backup arriving at the scene.
Yes, and their primary job is containment. The astrocytes undergo hypertrophy. They swell up and rapidly multiply, gradually accumulating around the electrode.
Sounds intense.
It is. They secrete dense extracellular matrix proteins to build a literal physical wall around the invader. This is what we call a glial scar.
Which isolates the probe completely.
Exactly.
And I want to highlight a statistic from the immunological study that perfectly illustrates the devastating cost of that scar. They observed rats implanted with these standard motor cortex microelectrodes.
A very standard test, yeah.
Right. And because of the blood-brain barrier damage, the persistent neuroinflammatory response, and the resulting glial scar, the rats experienced a 527% increase in the time required to complete a fine motor task.
A 527% increase. That is a staggering loss of baseline biological function caused entirely by the implant.
It's devastating. The very device that is engineered to theoretically help restore motor function is instigating the exact tissue damage that destroys motor function.
The irony is brutal.
Plus, that scar tissue acts like a dense biological insulator. It physically wraps around the electrode, pushing the healthy neurons further away and blocking the electrical signals.
Which is the death knell for a BCI.
Within a few months, the neural recordings degrade to static, and the device becomes completely useless.
If we connect this to the bigger picture, solving the brain-computer interface problem isn't a matter of building a faster computer processor or writing a better algorithm.
It's a biology problem.
Exactly. It is fundamentally an exercise in tricking the human immune system. You cannot establish a functional, long-term BCI unless you can convince the host brain that the hardware is supposed to be there.
Which brings us to the first massive engineering hurdle, making the hardware itself less like a dry piece of spaghetti.
Yes, exactly.
Here's where it gets really interesting, because the innovations outlined in the Material Science Review read like absolute alchemy.
They really do.
If stiff electrodes cause continuous micromotion trauma, engineers decided to radically redesign the physical materials to behave mechanically like the brain tissue itself. We are moving entirely away from solid metal wires.
Right. One of the most fascinating inorganic approaches is the transition to room temperature liquid metals, specifically utilizing gallium-indium alloys.
Liquid metal, like literally fluid.
Literally fluid. Instead of a rigid metal pin, imagine a conductive fluid encapsulated within a highly flexible polymer microchannel.
Okay, I'm picturing that.
Because the conductor is a liquid, it possesses extreme compliance. These liquid metal electrodes can be stretched to 200% of their original length without any loss of electrical conductivity.
That is wild. So as the brain pulses with blood flow, the electrode just naturally flows into forms right alongside it.
Precisely. The mechanical strain is distributed rather than localized. They're also achieving similar results with carbon nanomaterial.
Like what?
Like multi-layer graphene hydrogels. Graphene on its own is incredibly strong, but researchers are chemically converting these microscopic sheets and suspending them in a water-rich hydrogel matrix.
So it's mostly water.
A lot of it, yes. When the brain tissue pushes against this material, the water shifts, allowing the individual atomic layers of graphene to physically slide against one another.
It's essentially a shock absorber made out of sliding atomic layers.
That's exactly what it is.
It absorbs the kinetic force of the brain's movement rather than transmitting that force into the surrounding fragile neurons.
And that sliding mechanism creates significant viscoelasticity, which drastically reduces the physical compression on the cellular environment.
No, I have to ask about the mechanics of the surgery itself.
Okay.
If engineers are building electrodes that are as squishy as tofu or literal liquid channels, how does a neurosurgeon actually drive them through the meninges and into the cortex? I mean, you can't push a wet noodle into tofu either.
It's a brilliant engineering dilemma, and the solution is what researchers are calling a biological Trojan horse.
A Trojan horse.
Yeah, utilizing silk proteins. Specifically, they're coating these ultra-soft probes in fibroin, which is a protein extracted from silkworms.
Wait, silkworms?
Yes. When silk fibroin is completely dry, its molecules form dense crystalline structures. It becomes incredibly stiff and rigid.
Oh, I see.
Which allows the surgical team to machine it to a microscopic point and implant it with high-fidelity precision.
But once it crosses into the brain...
The brain is a warm, highly aqueous environment.
Very wet.
Exactly. The surrounding bodily fluids immediately begin to penetrate the silk coating. The water molecules physically break the hydrogen bonds, holding those rigid crystalline structures together.
So it literally melts.
Kind of. Within minutes, the silk plastifies, swells, and softens dramatically. adapting completely to the mechanical compliance of the wet brain tissue. It uses the environment's own moisture to change its physical state.
So let's play this out. If we successfully implement these materials, say we get the elasticity and the mechanical texture completely matched using silk or liquid metals, and the physical trauma drops to 0.
Okay. Does the immune system just ignore it? Are we in the clear?
You might think a perfect physical match would solve it, but the mechanical mismatch is only half the battle.
Really?
Yeah, the body's microscopic sensors don't merely detect physical trauma, they sample the chemistry of their environment.
Oh, right.
Even if the probe feels exactly like a neuron, the moment a microglial cell brushes against a synthetic polymer, its surface receptors recognize a non-self molecular pattern.
So the biological bouncer still spots the fake ID.
Perfectly said. The biological bouncer spots it, and the alarm cascade triggers. We don't just need a physical disguise, we require sophisticated chemical camouflage.
Because the immune system is sniffing the air, not just feeling the bumps. So how do we actually bypass that chemical alarm system?
Historically, the field relied on blunt force. The immunological literature shows that early researchers attempted to suppress the brain's immune response by flooding the implant site with broad-spectrum drugs.
Like what kind of drugs? We're talking about heavy corticosteroids, like dexamethasone, or broad-spectrum antibiotics like minocycline, which globally inhibits microglial activation.
Which seems incredibly reckless. Using broad-spectrum antibiotics to stop localized brain inflammation is like shutting down the power grid of an entire city just to silence one burglar alarm.
That is the exact danger. You are stripping the brain of its ability to defend itself against actual pathogens.
Did it even work?
Well, to be fair to the early research, using minocycline did temporarily increase the signal-to-noise ratio and prolong the lifespan of the electrical recordings.
But at what cost?
Exactly. Long-term systemic use carries severe metabolic consequences. For instance, chronic minocycline administration can cause the drug and its breakdown products to accumulate in the tissues.
And what does that do?
It leads to permanent hyperpigmentation, literally turning the skin and internal organs a blue-gray color.
Which is a terrifying side effect just to keep a neural chip online.
Absolutely terrifying.
And practically speaking, the sources point out a fatal logistical flaw here. Once a drug-eluting probe dispenses its payload, it's virtually impossible to reload it non-invasively.
Right, you'd have to do surgery again.
Yeah. The reservoir empties, the drugs fade, and the immune system immediately wakes back up to resume the attack.
Which is why modern BCI research has abandoned the broad-spectrum approach in favor of highly targeted immunomodulation.
Targeted how?
Instead of paralyzing every microglial cell, they are isolating the exact biological pathways that drive scar formation. A prime example from the research is the targeting of an enzyme known as caspase-1.
Walk us through what caspase-1 actually does in this context.
Sure. Caspase-1 operates as the executioner enzyme within something called the inflammasome, which is a multi-protein complex inside the cell that detects danger.
The inflammasome.
When the cell panics, the inflammasome activates caspase-1, and its entire job is to take dormant, inactive cytokine precursors and cleave them into their active, highly inflammatory forms.
So it triggers the alarm.
It essentially cuts the molecular lock off the pro-inflammatory signals that cause the glial scar.
So if you introduce a small molecule drug specifically designed to inhibit just caspase-1,
you successfully stop the maturation of those exact inflammatory cytokines, halting the scar formation process.
But you leave the rest of the immune system alone.
Crucially, yes. You don't turn off the cell's broader ability to fight off a bacterial infection. They're also having success targeting MCP1, which is a chemoattractant.
What does that one do?
When injury occurs, MCP1 acts as a chemical beacon that diffuses outward, enters the bloodstream, and recruits peripheral immune cells from the rest of the body to breach the brain.
Oh, it calls for backup from outside the brain.
Exactly. By blocking the MCP1 receptors, that chemical gradient is neutralized. The backup never arrives, and the localized inflammation is massively contained.
It's precision intervention. But the materials science review highlighted an entirely different strategy that blew my mind.
The non-stick approach.
Yes. Instead of using drugs to tell the immune system to calm down after it spots the debris, engineers are designing hardware that stops the biological debris from accumulating in the first place. It's essentially a microscopic non-stick pan approach.
Yes, the integration of anti-fouling surface coatings. This targets the very first step of the immune response.
How does that work?
When a foreign object enters the blood-brain barrier, blood serum proteins instantly absorb, or stick, onto the surface. It's this protein layer that the immune cells actually recognize and bind to.
So the cells don't see the probe, they see the proteins stuck to it.
Exactly. If you prevent those proteins from sticking, you essentially render the probe invisible to the cellular centers.
And to achieve that invisibility, they are utilizing a biological anti-adhesion glycoprotein called lubricin.
Lubricin is remarkable. It's a molecule naturally found in your joint fluid that provides near frictionless movement.
Like WD-40 for your knees.
Right. Engineers are pairing this with synthetic amphiphilic polymers, like sulfobutane. What these molecules do is tightly bind water molecules from the surrounding tissue, creating an incredibly dense microscopic hydration shell around the electrode.
It builds a literal force field of water.
Exactly. When a rogue protein or a fibroblast approaches the probe, it hits this densely packed hydration layer.
And it just bounces off.
From a thermodynamic perspective, it takes too much energy for the protein to displace the water molecules, so it literally just slips off. It cannot find purchase to trigger an immune reaction.
That is so clever.
The mechanical benefit is also profound. When researchers treated a silicon probe with this anti-fouling lubrication layer, the friction generated during the surgical implantation dropped by 86 times.
An 86-fold reduction. That means the acute physical trauma, the immediate shearing and tearing of cells upon insertion, is mitigated right out of the gate.
It is elegant prevention rather than continuous biological combat.
So let's take stock of where this leaves us. We've replaced the rigid hardware with liquid metals and softening silk.
Check.
We've cloaked the device with water-binding polymers so proteins slide off, and we're using highly precise molecular inhibitors to silence the specific inflammasome alarms.
All true.
We've successfully hidden the device. So what does this all mean? Can we finally plug in and download a new language?
Well, we aren't quite there yet.
Of course not.
What's fascinating here is that evading the immune system only ensures the device's survival. It does not ensure its utility.
Right.
For a brain-computer interface to function, the neurons in the surrounding cortex actually have to connect to the device. They need to establish a stable, high-fidelity electrical relationship.
Right, the brain has to actively plug its own organic cables into the artificial ports we're providing. How do we coerce a neuron to do that?
We achieve it by speaking the physical language of cellular growth. This introduces the field of micromorphology, or microsculpting.
Yeah, sculpting.
Yeah. Neurons navigate their environment entirely through topographical cues. At the tip of a growing neural axon is a growth cone, which is covered in specific adhesion molecules called integrins.
You can think of those integrins as microscopic hands reaching out, physically feeling the microscopic textures around them to decide which direction to grow.
Precisely. If engineers introduce a perfectly smooth, flat metal wire, even if it's biologically tolerated, it offers zero navigational guidance.
It's just a blank wall to them.
To a neuron, it is a featureless desert. They will either wander randomly or ignore the surface altogether.
Yeah.
But if we change the topography, the behavior changes.
How small of a change are we talking?
We are talking about utilizing ultra-fast femtosecond lasers or deep reactive ion chemical etching to carve nanoscale grooves, staggered columns, and biomimetic porous scaffolds directly into the surface of the electrode.
Wow.
Structures measured in billionths of a meter.
And when the integrins feel those specific nanoscale grooves?
It triggers a process called contact guidance. The physical shape of the nanostructure mechanically forces the internal cytoskeleton of the neuron to align with it.
So they literally follow the path.
The neural axons will lay themselves down inside those artificial channels, following the precise path carved by the engineers.
It completely reframes what the implant is. It stops being a foreign object and becomes a trellis.
A trellis, exactly.
Just like a garden trellis that a climbing vine naturally wants to wrap around and grow up. By microsculpting the electrode surface, we are building roads that specifically invite the neurons to intertwine intimately with the hardware.
And that intimate physical wrapping is what ensures the electrical signals are transmitted with crystal clarity.
Without having to blast high voltages across a dead gap.
Right. That intimate wrapping represents the ultimate interface. We are transitioning from crude devices that simply sit inside the brain and attempt to eavesdrop on the electrical chatter from a distance to architectures that actively invite the brain to grow into them, forging a seamless biosynthetic network.
It is an unbelievable shift in perspective when you look at the entirety of the research.
It really is.
To summarize for you listening, we started out analyzing this problem as an engineering sledgehammer, shoving rigid, brain-damaging metal pins into the softest tissue in the human body, triggering massive immune reactions that effectively deafened the device.
The dark ages of BCIs, essentially.
Right, but the future of BCIs is incredibly elegant. We're looking at liquid metals that stretch effortlessly with your pulse, silk proteins that dissolve and soften on command, microscopic water force fields that deflect proteins.
Don't forget the pinpoint chemical interventions.
Exactly, the targeted drugs that only snip the Caspase-1 alarm wire, and laser-etched trellises that actively invite your brain's growth. We aren't fighting the biology anymore, we are learning to seamlessly merge with it.
This raises an important question, actually. One that pushes past the biology and the engineering, diving into the profound philosophical implications of this success.
Okay, I'm ready.
If we truly perfect this, an electrode that matches the brain's mechanical texture flawlessly, completely evades the chemical bouncers of the immune system, and structurally integrates directly into our neural scaffolding, at what point does the brain start recognizing the machine as self?
Oh, wow.
Think about the mechanics of the mind. Our brains maintain a constant internal map of our physical bodies. We know precisely where our arms and legs are in space, even with our eyes completely closed.
Right, proprioception.
Exactly. That physiological sense is called proprioception. If a brain-computer interface bends, feels, and signals in perfect harmony with your organic neural circuits, future humans might possess a genuine biological sense of proprioception for their digital limbs.
That is mind-bending.
You might be able to feel your connection to an external hard drive or the position of a robotic prosthesis as intimately and naturally as you feel your own biological fingers. If the interface becomes truly seamless at a cellular level, where exactly does your biology end and the computer begin?
That is a staggering thought to leave on. If we manage to perfect the scaffolding and the chemical disguise, the brain might just adopt the machine as its own native tissue. Thank you for joining us on this deep dive. Keep questioning, keep exploring the boundaries of the human mind, and we'll see you next time.
Yeah, go check out BioniChaos.com. Eventually, everything we make is available on this site, open source, free of charge, no login. All these simulations and things are available for you. The source code is on the page. Check it out, provide your feedback, and I'll see you next time.